One thing that we had when we switched, philosophies here, and we used to do 18 different amino acids, we had questions about, well, are we sure that, a couple of different amino acids are better than applying all 18? And so, at the end of the day, what we're doing here and where the opportunities are is that these amino acids that we're applying, and in the case of aspartic acid and glutamic acid, we call them upstream amino acids because what happens is through different aminotransferase enzymes, they can turn into other, quote, downstream amino acids. And so instead of applying 18 amino acids and just hoping for the best and saying, hey, we know you need histidine and lysine and methionine and all the other great amino acids out there, now we're letting the turf decide, okay? So the turf can take glutamic acid and it can turn it into 3 different amino acids in one step. And from those, it can turn into, glutamine can turn into histidine, and ornithine turns into eventually arginine. So that's great. Histidine is important for zinc and nickel accumulation in plants. That's wonderful, okay? But instead of just applying histidine, we're applying a high amount of glutamic acid, and we're letting the plant decide. The plant's going to send signals within itself to say, hey, you know what? I need to work on my zinc and nickel accumulation. I'm going to transfer some of this glutamine and turn it into histidine, right? Or arginine, right? It's important nitrogen storage and transport. If the plant and turf needs to store nitrogen, it'll turn it into arginine. Great. Proline, we're going to talk about proline a lot here in a little bit. We talk about anti-stress, but it's an osmolyte in the vacuoles, meaning that it's loaded in the vacuoles, and it helps the water stay in the vacuoles, and the vacuole will maintain its shape, which again, turf's composed of 80% water. That's really important stuff. But again, we're allowing the plant to kind of decide this and what it's doing. Same for aspartic acid. In one step, it can turn into asparagine, methionine, lysine, and threonine, and then some of these amino acids can turn into others. So again, it indicates nitrogen status in the plant with glutamine. There's a balance there. Great, okay? But we're not going to apply it, and we're going to let the turf decide if it needs it. Methionine, building up signal transduction in plants. It helps the plant to communicate with each other. It's kind of an interstate within the plant. Methionine plays a role in that. Lysine can turn into glutamic acid. It also can turn into dehydrin proteins, which helps the plant go through these stressful times, especially when you talk about water concerns. And threonine, it helps regulate the levels of pyruvate. It's involved in the energy production in the plant. All great things. In the past, we would have just sprayed all these amino acids out in various concentrations, and it was a great story. But now we've got the science to back it up, and so we're sitting there, and you're probably asking yourself, and if you haven't done it yet, you might later. How do we know, George? How do we know that glutamic acid and aspartic acid turns into these different amino acids, or turns into chlorophyll, or turns into any of the structures that we've talked about? And this is research we're going to continue to do in 2017 that we've already done. We're taking isotopes of these amino acids, okay? So we're adding an extra proton to your nitrogen or an extra proton to your carbon. The carbon has 12 protons, and nitrogen has 14 protons. And what this allows us to do is to be able to track this. And when we spray these isotope amino acids on the foliage of the turf in the greenhouse, and then we harvest that turf, we're able to take it to the metabolomics facility, and we're able to actually track where did that carbon-13 or nitrogen-15 amino acid turn into. Did it turn into glutamic? Did it turn into chlorophyll? Did it go into proline? Did it do other sorts of things? And we can track it down to that level. And that's exciting. For us, we're able to check the uptake rate for these different amino acids in optimal growing conditions, but we're also able to see the metabolic state when there's different environmental conditions. So when the conditions are optimal, the plant says, hey, you know what, I'm doing good. I'm just going to make more chlorophyll, or I'm going to bank this into proline, or I'm going to turn this into lysine, right? In drought stress, the plant may say, you know what, I'm going to take this glutamic acid, I'm going to turn it into as much proline as I can, and as much gamma-amino butyric acid as I can. I'm going to load it up. So there's different signals that happens, and the plant can use this amino acid gift, which is basically what it is, in different ways. So I just wanted to make you guys aware of it, say, hey, we're doing this, we've done it, we've got early data in our hands now, and we're going to continue this in a big way. And this is a unique way to look at it. I think in the past, when you look at what other people have done, they go to a university and they go to the turf guy and they say, hey, Dr. So-and-so, why don't you spray this out in replicated trials, and let's see if it changes color. Let's see how it survives the summer. Let's see all this stuff. And don't get me wrong, we're doing that. But to me, the question is why? Why is this happening? And to do that, you've got to break it down into metabolic physiology. And that's the exciting research that we're doing and we're going to continue to do.